welcome to some video notes. We are going to be walking through the process of gel electrophoresis. So you'll be watching through this video, and then we will be actually using some of the different instruments that we see in this video here to then get comfortable with using what's known as a micropipette. And then in our next week, we will be doing gel electrophoresis in class to look at the different DNA patterns that are created based off of the different DNA makeup of each individual. So I can't show you this video because of copywriting purposes. What I can do is I'm going to link it on Schoology so you guys can watch the video. You can either pause this one, hop over to the gel electrophoresis video, and then hop back to the video notes, or you can save it to watch at the end. It's up to you, but I definitely recommend watching it because this will help the process that we then do in class make a lot more sense. So gel electrophoresis in general allows us to create DNA fingerprints based off of each individual's unique DNA makeup. And it's actually a great way to visualize the actual differences in each individual's DNA, which is incredible, right? We went from not knowing what DNA looked like to now being able to not only understand more of its structure, but actually see it doing these kinds of processes. So very, very, very cool. I always love to see how science just continues to develop. So in terms of the gel electrophoresis process, there's 4 main steps that occur. So the DNA has to be extracted from the cells, and then you'll introduce what's known as restriction fragments. So these are essentially different enzymes that will look for specific DNA bases and will cut them at those locations. And in cutting then the DNA at those specific locations, we can then do step 3, which is amplification, where we then just make a bunch of copies of all of those little sections of DNA. And making a bunch of copies then allows us to then visualize those DNA fragments in step 4, which is gel electrophoresis. And what a gel electrophoresis does is it actually moves the DNA fragments that we cut up using those restriction enzymes and then amplified using a process known as PCR. We can look at those fragments and they'll move depending on the size of the fragment. So let's say, for example, the chunk of DNA that I have for one individual is 7 or 8 different nucleotide bases long. The next individual, because everybody has a different sequence, might have 9 or 10 nucleotides in that sequence. And so based off of the size, the shorter the fragment, the faster it's going to move. And so that means then the faster it'll move down through the gel electrophoresis. Once again, this conversation will make a lot more sense if you watch the video on the last slide. So if this is kind of confusing, I would go watch that video and then come back to this one. But essentially, the length of the fragment decides then how quickly it can move. If it's short, it's going to move a lot faster, which means it'll end up further down our DNA fingerprint. If it's longer, then it's going to move slower, which means it'll stay near the top. So in terms of each step, looking a little bit more in depth into these processes, the first step is extraction. And this is what we did when we did the strawberry DNA lab. We literally extracted the DNA from it. If you remember, it looked kind of like spiderwebby saliva-ish. And that's essentially what we do for gel electrophoresis. We want to extract the DNA out of the cells. And there's different kinds of detergents and solutions that we can do to do that. In our strawberry DNA lab, we just use soap and salt. So for the next step, we use restriction enzymes. Restriction enzymes essentially look for certain patterns of DNA bases, and we call those restriction sites. And then they cut the DNA at that specific location. So for example, this restriction enzyme will cut the DNA at this G and this E, or this A here. So this is where it's going to cut. We'll have an entire strand of DNA. This enzyme is going to look for every single part of our DNA that has this pattern here, and it'll cut it at the same exact spot. Same with this enzyme down here. You can see that it will cut at a CG location, and it'll just look through our DNA for all of these different little codes, DNA bases, and it'll make that cut. And so this forms different sized fragments of DNA based off of each individual, because once again, all of our DNA is different. And so I might have a super long fragment while somebody else might have a really short fragment. And then once again, the length of our fragments is going to create different DNA fingerprints because of how fast and slow they move. And then, so these restriction enzymes are really cool because they were actually produced by bacteria to defend themselves against invading viruses. So if we remember our COVID vaccine project, this is all coming together, everything we've done this school year. The COVID vaccine project talked about how these viruses literally inject their DNA into bacteria, and they turn the bacteria into virus-producing factories, essentially. The bacteria use these enzymes to protect themselves. If that virus tries to inject its DNA and turn the bacteria into a factory, the bacteria uses the restriction enzymes and says, hey, no, chop, 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 chop. I don't want to become a factory. Please yeet yourself out of my cell, which is cool because then we're able to use this for science. So in terms of amplification, this is where we use that PCR, which stands for polymerase chain reaction, and this can be used to amplify pieces of DNA. So in practice, essentially what we do is we look for that region of interest within the DNA sample, and then what we'll do is something called denaturization, which essentially separates the two strands. And then with those two strands, we can introduce the enzyme DNA polymerase, which then makes copies of all of the bases, which is known as elongation. So this should sound a little bit familiar for DNA replication. It's the same enzyme, DNA polymerase. So it'll make copies of each strand, and then billions of those copies are then generated to then be able to use those specific parts of DNA for further analysis via gel electrophoresis. So let me just go over this one more time shortly. We have our DNA sample. We said this was the part we want to focus on. So then we split the DNA into two strands, put down some primers so that the DNA polymerase can make copies, and then we just make tons and tons and tons and tons of copies so that we can better visualize the way that our DNA is moving through the gel. And then finally, for gel electrophoresis, we load the DNA into the wells at the top of the gel. And this gel is known as agarose gel. It is porous, kind of like a sponge, and that allows the DNA fragments to move, but they have to move through a specific way, so that's why shorter fragments move a lot faster than longer fragments. And so once you have the DNA loaded into each of the wells, then you'll turn on an electrical current because DNA is negatively charged. It's going to move down towards the positive end of the current that we're running through our gel. So then once you have completed your electrophoresis, you can turn off the charge, and then you can look to see the DNA fragments that you were able to then separate, and that creates your DNA fingerprint. So the largest fragments end up on top, the smallest fragments end up towards the bottom, closer to the positive end, because we're going to move, if this is negatively charged DNA, it's going to move towards the positive side, and it moves depending on size. Larger is slower, smaller is faster. So that's pretty much all the information that we need to know about electrophoresis. This is the process that we're going to be doing in class next week. This week, we're going to just be practicing with those micropipettes, getting used to taking up and dispensing out those solutions. So if you guys have any questions along the way, of course, I'm always here to help you guys out. Otherwise, I look forward to seeing what you create. Thanks, guys.